lesson we are going to study about development of seed. So in our previous classes, that means in our previous one classes, we already discussed about some of our post-fertilization event, and we know, that means the gist of your post-fertilization event, that some type of changes are takes place after the fertilization. So the changes are basically the ovary changes to fruit, that means the ovary is changes to form your fruit, and the ovule is changes to form your seed, and the integuments, that means the bitechnic integuments, is formed to your seed coat. That means this is the basic changes occur in your post fertilization event, that means after fertilization these types of changes are takes place. So these types of changes is most common to a seed to form a fruit. So if we study the total overall topic of your development of seeds, we already discussed it overally in your morphology, and as we are studying sexual reproduction in flowering plants, so the ultimate point, that means the ultimate point of sexual reproduction in flowering plant is a fruit or the seed, that means development of a seed or a fruit. So I just give you a basic concept about the development of seed. So we just overlook the structure of a seed first. So if we saw the structure of a seed, it may be a bean seed, it may be a mango seed, it may be a gram seed, it may be some other seeds also. It consists of a seed coat. That means the seed coat is replaced by your integuments. That means the integuments changes to form your seed coat, and the seed coat is again divided into two types. The outer part is otherwise known as your testa, and the inner is your tegmen. That means the seed coat is again divided two parts. That means the outer seed coat is otherwise known as your testa, and the inner part is otherwise known as your tegmen. So this is the outer part. Nextly, the seed is consists of a hilum, and the hilum is changing. That means according to the seed, the hilums are many types. That means if you consider that this is your bean seed, so in the bean seed it is in the middle part. That means it is the hilum. And if we talk about this gram seed, this is your gram seed, so the hilum is here, and next to the hilum the micropyle is there. So the micropylar is there. So the micropyle and next to the hilum, and it is also the testa and the tegmen are also there. The seed goes in there. This is the structure of your gram seed. So basically, the seed is consist of two types of layer. One is your outer testa and inner tegmen, and consist of a hilum part and in embryonal axis. That means in embryonal axis, this is your embryonal axis. If we open the seed, that means it is one side of the seed. We just draw two sides of the seed. It consists of two types of cotyledons. That means two cotyledons, and cotyledon is mainly help to nourishing. That means it is the reserve food material in it. And in monocotyledons it is thin and papery in nature, but in dicotyledons it is thick and fleshy in nature and consists of a number of nutrient medium in it. So here is the cotyledon, and nextly the upper, that means this is the embryonal axis, and the embryonal axis, the parts of the embryonal axis we already discussed in your formation of your endosperm. That means your after the endosperm formation, we just discussed all these things. I just recap all these things. So nextly the plumule formation and the radicle. Radicle is the baby root. That means the root formation is there and the shoot formation is there. So in these things, that means in this manner, the total seed is going to develop. That means it is the developmental process of a seed. And after the developmental process of the seed, I just recap what are the classification of the seeds. So in your previous classes you also know about that. I just recap it. So your seed is mainly consist of, if you talk about a basic seed, it consists of mostly three things. That means seed coat, which are found from the integuments. That means the seed coat is mainly formed from the integuments. Nextly, the embryo. Embryo means what? The embryo means the baby plant. That means here the plant is formed from the embryo. Next is the cotyledon. Cotyledon means the reserve food material. That means the seed is consist of basic three things. One is your seed pod, another one is your embryo, next one is your cotyledon. According to this cotyledon, that means according to this reserve food material, we just divide it into two types. One is your dicotyledon, another one is your monocotyledon. Dicot means your two cotyledon, and monocotyledon means one cotyledon. So, if we talk about the dicotyledon, it again divided it into two types. In endosperms, that means the nutritive tissue endosperm. When I told you about the endosperm, I told you a point that it is albuminous in nature. In some cases, it is ex-albuminous in nature. So, it is divided into two types. One is your albuminous. Albuminous means endosperm. It consists of some types of endosperm. When the total formation, that means when the total embryo is formed, some type of nutrient medium are left in it, and the left nutrient medium containing plant is always known as your albuminous. That means the seed is always known as your albuminous. The most common example is your castor. And next to the endospermic is opposites. That means endospermic and non-endospermic, albuminous, ex-albuminous. So the opposite means the totally opposite thing. That means here the nourishment is there and the nutrient medium is left, but in this case no nutrient medium is left, and the nutrient mediums are mainly set in your cotyledon. That means from the cotyledon the plant is going to that means the plant is started its developmental structure. So in ex-albuminous, the most example is your pea, gram, beans. That means the pulses are the best example of your ex-albuminous. In the other hand, in the case of monocotyledon, I told you about, that means your non-endospermic, and another one is your endospermic. Non-endospermic, in some cases thin and papery cotyledons are there, and the most common example is orchids. And the endospermic, the most common example, that means I know that you all know that mostly the nutrient medium are commonly used of in this case. That means in this endospermic condition, mostly the nutrient mediums are used of, but completely used up, but in some cases some are left. The things which are left is considered over your non-endospermic in monocot and albuminous in your dicot. But which are used of is kept under the endospermic, and endospermic are mostly the cereal grains. That means your rice, maize, wheat. These are some of your example of your endospermic. So this is the basic classification of your seed, and this is the basic diagrams of your seed. I again repeat, the seed is developed. That means the developmental structure is mostly takes place in three stages. One is your ovary changes into your fruit, and ovule changes into your seed, and integument changes into your seed coat. We already discussed it completely in your post-fertilization changes. Nextly, we just overlook the structure of your seed. So, in structure of your seed, the outer layer, that means the outer seed coat, which is formed from the integument, is divided into two types. That means the outer layer is always known as your testa, and the inner layer is always known as your tegmen. That means two types of layer are already formed. Nextly, the middle part, that means the point is always known as your hilum, and there is a axis in between there. That means between the cotyledon there is a axis in it, which is always known as your embryonal axis, and we already covered the embryonal axis also. The upper part of the embryonal axis is consist of your plumule, and the lower part is consist of your radicle. From the radicle, the root part is developed. From the plumule, the shoot part is developed, and the cotyledon is just the reserve food material. It is nothing but it is just the reserve food material. It reserves the food and gives the nourishment when the developmental structure. That means when the development is takes place, when the germination is takes place, and the time the nutrient medium is transferred from the cotyledon. So, according to this basis, we just divide it into its two parts. That means your dicot and monocot, and dicot is further divided into two types. That means one is your albuminous or endospermic, another one is your ex-albuminous or non-endospermic. In albuminous, I told you the example of your castor, and in ex-albuminous, I told you the example of your most of the that means your pea, gram, bean, etc. And in monocot, it is again divided into two types. Non-endospermic consists of your peppery thin your structure, and the most common example is orchid. And in other hand, the endospermic seed, the most common example is your number of your cereals like your rice, maize, wheat, etc. So these are the common differences and the total developmental structure of your seed. Nextly, the significance of this seed. So we already studied in the significance of the seed. That means the seed is the main structure of your formation of a plant. That means from the seed only the total plant is formed. So seed can be used as food throughout the year and also to raise crop next year. That means if we talk about the grains like your maize, wheat, rice, etc. So these are stored, and these are the mostly the common food source of our human beings. So these are raised of crops, and we just preserve it year by year and in cultivated year by year. And the most common varieties are we also found, and most that means the seeds are able to adapt one generation to another generation. That means the adaptability capacities are takes place. A single evolution is also takes place from the seed. That means from one generation to another generation. So in second point we discuss about the seed store enough food materials, either endosperms or cotyledons, to nourish the seedlings. That means at the time of seedling, that means I told you over here that if we talk about this types of seed, it consists of cotyledons, and in cotyledons it consists of number of nutrient medium for nourishing the plant. That means when the seedling is takes place, at that time it gives, that means the cotyledon is only gave the food material to the developing structure. That means from this only the seedling is takes place. So seed stores enough food material in it. That means these seeds, that means you may take your pulses, you may take your rice, you may take your wheat. All these grains are consist of nutrient medium in it. Nextly, seed have adaptations for dispersal to new habitats. New habitats means a new area. That means you can transfer a number of seeds from one stage to the another stage. That means one area to another area to developmental structure. That means there must be a dormancy period also there. And all these things are expressed. That means seed germination and the dormancy period. According to this, a number of things are developed in a seed from one generation to another generation. You may take as the evolution of your seeds are also expressed. So this is the changing of your habitat and have responsible for distribution. It can distribute it from one area to another area. That means you may take as example of your rice variety from one area to another area. It may be transferred from one area to another area. Seedlings are also takes place, and germination takes place from one area to another area. That means the changing of the habitats also takes place. That means the seeds are adapted from one area, and it can also adapt from another area. It may be grow easily from various type of environmental conditions. So these are some of your significance of your seed. That means dormancy, you all know about the dormancy period of the seed. So these are the some of your common examples and some of the significance of your seed and development of your seed. Hope this class is helpful to all of you. Thank you.